Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to everyone. Okay, a warm welcome to all participants joining us today, and both here at Seminar Hall Room as well as virtually via Zoom as well as Facebook Live. To our Muslim participants, I would like to extend a warm special greeting of Salam Ramadan Kareem. So may this blessed month bring peace, reflection, and abundant blessing to all of us. Welcome to INBIOSIS Hybrid Seminar Series 2, organized by Institute of System Biology, Universiti Kebangsaan Malaysia. My name is Murni Nazira Sarian, and I am honored to serve as the moderator for today's seminar. So we are delighted to see such wonderful turnout today with participants joining us both physically and virtually from various institutions and backgrounds, all sharing a common interest in advancing knowledge in system biology and biomedical sciences. So today's seminars features a very exciting and cutting-edge topics entitled Turning Tumors into Turncoats, Developing Off-the-Shelf Living Drugs and Immunogenic Suicide System for Solid Tumors. So this seminar highlights innovative approaches in modern cancer therapy, particularly the development of engineered biological systems that can be programmed to recognize, target, and eliminate tumors. So before we proceed, allow me to introduce our speakers today. Dr. Chen Siong began his academic journey at University of Malaya, where he obtained his bachelor's degree in genetics and molecular biology, followed by a master's of science in pediatric oncology. Then he later pursued his PhD in neuro-oncology at University of Queensland, Australia, where he focused on advancing understanding of brain, tumor, and cancer biology. Following his doctoral training, Dr. Chen joined Brigham and Women's Hospital, Harvard Medical School as postdoctoral fellow, where he spent 4 years conducting advanced research in cancer biology and therapeutic development. He subsequently continued his academic career at Harvard Medical School, where he served as instructor for nearly 3 years, contributing to cutting-edge research at intersection of oncology, immunotherapy, and engineering. Okay, without further ado, I would like to welcome Dr. Chen Siong to share his expertise and research insight. Welcome.
Hello, everyone. Thank you for the warm welcome by Dr. Murni Nazira. So maybe she didn't mention that, but she is actually my classmate when we were in the secondary school. So salam sejahtera to everyone. And then I know now it's puasa, so I will try to keep it short and sweet, and then see how it goes. So just some brief introduction about the work that I'm doing. So basically, our lab is focused on cell-based therapy, developing the cell-based therapy to treat the kind of like the gill tumor. So like the GBM glioblastoma that we are trying to treat. So my focus and my presentation today will be mostly on this brain cancer that we are working on. So before I start, I'm going to share my slide. So before I start, I would like to share with you a video that just to have a brief introduction on what we have done. So this video basically is a news from Boston. So when our paper just published, then they right away pick up by this news agency, and then they report it in the news. So I think it's a good introduction to have what we have been doing. So I'm trying to see whether it can play well. So if you cannot hear anything, please let us know. No? Okay. So I might be trying to use the microphone from here to play from my speaker. Is that okay? Huh? Not sure whether here. Thank you. Well, let me try again. Okay. So now let's try. Can you hear that? No.
Tonight, local scientists have found a way to fight cancer with local scientists have on the Health Watch.
Huh? Oh, you cannot hear from here. How? If I play from here without this,
This online cannot hear. Yes, okay. You have increased the volume, right?
so it is the highest. Or you can
One.
Tonight, local scientists have found a way to fight cancer with cancer. Dr. Malika Marshall is here.
Fine.
On the Health Watch tonight, local scientists have found a way to fight cancer with cancer.
So online can hear? Okay. So my apologies. So let's try again.
On the Health Watch tonight, local scientists have found a way to fight cancer with cancer. Dr. Malika Marshall is here, and we're specifically talking about brain cancer.
That's right. These were scientists actually at the Brigham who are using what they call living brain tumor cells to stimulate a patient's own immune system to destroy tumors and to actually prevent cancer. So they say these living tumor cells will travel long distances across the brain to seek out fellow tumor cells, similar to sort of homing pigeons returning to roost. To take advantage of that behavior, they have engineered these tumor cells to release a tumor cell killing agent, as well as factors that allow the body's immune system to spot and tag and remember tumor cells in the future. They found the approach was both safe and effective in mice models, and what they want to do now is develop a cancer-killing vaccine for use in humans with brain and other solid organ tumors.
Sounds like an amazing discovery, right? All right.
Yep, that's it. So shall I put the jack back? Oh, it's okay. Okay, so we'll continue. So like what this short video described. So my work basically kind of like trying to turn the cancer cell itself as the therapeutics and then trying to treat the cancer. So in this case, what we are trying to do, so in the kind of like brief introduction, because our main focus why we want to use the cancer cells is just like the idea that we know that a lot of like let's say smallpox, we will use the vaccine. we inject the disease itself, the attenuated disease itself, and then we put it back to the body to train our body, to train our immune system so that when they encounter the similar type of disease, they can actually reject it, they can eradicate it. So the idea is kind of like derived from there. So a little bit of the about the what is a cancer vaccine. So a lot of people will say, what kind of vaccine that you can have? Because cancer, you will never know when you are going to get the cancer. So what we are trying to do with the cancer vaccine. So from the cancer vaccine itself, the terminology itself is that they can be divided as two parts. One is the preventive. So you can see from here that the preventive is that when you inject this kind of like a vaccine, it can prevent the tumor from growing. So one classic example is the HPV vaccine. So HPV vaccine is because it's more viral derived cancer. So this can be prevented. So you can use something to the disease, the foreign things to actually induce it in our immune system so that it can prevent the HPV cancer from being developed. And then the other one is called more therapeutics. So what it can do is that it can train the immune system to target the tumor cells. So you already have existing tumor cells in your body, but this cancer vaccine term is kind of like trying to train the immune system to kill them. So there are multiple types of cancer vaccine. So basically from here, you can see we can categorize them into 4 types. One is the predefined and personalized. So you kind of like know what kind of target that you want to target. So you use, for example, you already have some peptide vaccine that designed to target certain tumor antigens. So, and then you know this patient is going to have these antigens. So for example, for breast cancer, we know that they are HER2 positive. So in this case, we can use the HER2 positive use the vaccine that they can target the HER2 positive cells. So this is more predefined and personalized. And the second one is the anonymous and ex vivo. So we don't know what they have. So we just kind of like try and error. We just put it in and then see how it works. So this is more like you are using the common tumor antigens. So because we know that most of the tumors that share the similar kind of antigens, but instead of personalize it, we already have a ready-made vaccine that we use it, that we modify it, and then we do it on the patient. And then the third one is you predefine, but it's a share. So this is more like more into you also know what you want to target. You already make them specialized. This vaccine, they are not targeting random antigens. They are kind of like doing they know what they want to target. So this is a C, and then D is anonymous and in situ. You basically know nothing. You just doing nothing and then see whether it's going to manage to train the vaccine. But in this case, our strategy for my whole research is more into this anonymous ex vivo. So we are trying to target more generally how the whole patient, how it is going to have the different patients should have similar kind of like vaccine that we will have. So you can see that there are cell-based cancer vaccine, viral-based cancer vaccine, peptide-based cancer vaccine, and nucleic acid-based cancer vaccine. mRNA vaccine belongs to here. Peptide-based is more into they use different kind of like peptide to induce the immune system. And then our focus is cell-based cancer vaccine. So just give a little bit of the history of using cancer cells as the vaccine. So in 1959, this Brigham couple, they are husband and wife, they have this idea. Why don't we just put the cancer cells, the whole tumor cancer cells, to really put it back to the patient to induce the immune system like other diseases? So, but they kind of, because it's a living cells, so what they did is that they attenuated them. So to make them inactive so that they cannot proliferate anymore, and then inject them to the patient. So the whole idea started from there. So after several decades of research, this kind of like proven, yes, they might induce some immune response, but it's not enough to really clear the tumor. Because as you might know, because when the tumor cells most of the tumors, not most, yeah, most type of the tumors are derived from our body. They derive from normal cells. So to really educate our body immune system to target something belongs to us is kind of hard. It's very hard to do that. So the other type, then there are this group that from Drenov that in 1993, the first clinical trial that he kind of like run is in 1993, is to accompany this attenuated whole tumor vaccine together with the expressing cytokine. So the cytokine that they chose is called GM-CSF. So they notice that when you're putting these tumor cells, the attenuated inactive tumor cells together with this GM-CSF, it can become a kind of like adjuvant to induce the immune system to help to have the effective immune system to target the tumor. So this kind of like also effective, but not effective enough. So in 2018, our group actually derived from here. So we would like to use the living as why not we try to use the living whole tumor vaccine in this case. So we have this idea why the attenuated whole tumor vaccine is not effective because they cannot kill the tumor themselves. They don't have the ability to target the tumor. They can only work as a vaccine to induce the immune system, which is kind of like passive. We want them to be also active and also passive. So in 2018, we proved that we can actually make this this living whole tumor cells as an active killer to kill the tumor cells. And then in 2023, we go even further because in 2018, we were only looking at the killing ability. We didn't incorporate the immune response yet. So in 2023, we also incorporate the cytokine that we mentioned from here together with the tumor targeting agent to make them bifunctional killing vaccine. So this was published in 2023. That's the time that this news that I show you guys about for this. After we publish this paper and then they have this news. So it's more into this bifunctional killer vaccine. So now I will move on to explain into this bifunctional killer vaccine that we developed. Why these therapeutic tumor cells? There are several features that we chose them. Why we chose them? First is because the self-homing ability. So these tumor cells, they like to go to tumor cells itself. So in this case, we don't really need to think about targeting targeted therapy because when we have these living tumor cells, when they travel in our body, they will go to they will go back to their own tumors. That's how why the tumors become bigger and bigger because all the tumor cells that are flowing in the blood, they will always go back to their original site. Or there's also the reason why there's a metastasis. When there's another lump of tumors start to establish, reside on a certain part of the organs, the other tumor cells will also go there. So the self-homing ability kind of like let us to, we can kind of ignore the part that we won't have the targeting problem in this case. So that it can solve the delivery challenge. The second one is the antigen shedding. So that's also why those other vaccine works because these tumor cells, they always shed the antigens. These antigens will be something to be trained, to be used to train the immune system to learn that this is the foreign cells in our case. So it can help to solve the tumor heterogeneity because we are using the whole tumor cells. If you're only using one particular antigens, you can only target those kind of cells. But we know that within the whole solid tumor, we have multiple different antigens, and then each of different cells, they have different characteristics. So the best idea is to put lump sum, the whole thing, just put in there, and then train the immune system to target everything. That's what we are trying to do. And then the best thing is that it's amenable to gene editing. So not all the cells can be edited genetically, but cancer cell is the best candidate because they are proliferating very fast. So very easy to edit them and then use them, and then they can still live life. They can still stay alive in this case. So these are the main thing that why we choose tumor cells as a therapeutic tumor cells. Because compared to what currently people using is a CAR T, they're using the CAR T cells to make the CAR. So we have the advantage to have the self-homing in this case, and also have the antigen shading. So that's two different things that these tumor cells has. So now briefly introduce what we have done for this bifunctional therapeutic tumor cell. We call it THTC. Therapeutic tumor cell, THTC. So from here, you can see that first we want to incorporate the directly killing ability to these cancer cells. What we choose is the interferon beta. So we notice that in this all in all these tumor cells, apart from not just GBM, glioblastoma, even for other kind of tumor cells, they overexpress this interferon alpha receptor 1. So they have this receptor. So that's why we choose this. So you can see that the analysis we did, you can see that the overexpressed in the primary, they also always express in the recurrent. We compare to the expression level in this case, the interferon alpha receptor 1, interferon alpha receptor 2, they are very high. And then we also compare them with those known markers for this GBM. You can also see that the expression of interferon alpha receptor 1 and 2, they are kind of like comparable to other kind of like markers compared to the commonly known markers compared to this interferon alpha receptor 1. So this kind of like tells us it's a good target that we can use because they are overexpressed. So we don't have the problem of toxicity that will target the other healthy tissue in the brain. So, and then we make these tumor cells to express the interferon beta. And then this can actually, interferon beta has been known that they can mediate the cell cycle arrest and apoptosis. Once this interferon beta binds to the receptor of the tumor cells, it will induce the apoptosis. But then we have another problem. Oh, sorry. So this is just to show the pathway that they go through. So we have another problem because if these therapeutic cells themselves, they also express interferon alpha receptor 1, meaning that if we overexpress the interferon beta, they will first kill themselves before they are killing others. So to prevent that from happening, we knock out the interferon alpha receptor 1 from these therapeutic cells first before we make them interferon beta positive. So this is kind of like we use the CRISPR technology to knock out the receptor from the therapeutic cells so that they become from sensitive tumor cells to become the resistant tumor cells. And then we also incorporate the GM-CSF that I mentioned. They have this GM-CSF can help the maturation of the dendritic cells, so which can help for the antigen presentation. We also incorporate this GM-CSF to make them also bifunctional, also have the immunomodulation. And in fact, interferon beta itself is also the immunomodulators. So you can see from here, the indirect effect from interferon beta is that it can modulate and stimulate the anti-tumor immune response. So you can see that it can improve the cytotoxicity of NK cells. It can actually suppress the function of MDSC. The most important thing is that it can help the antigen, it can help the maturation of dendritic cells for the antigen presentation. So this is kind of like can to synergistically work together with GM-CSF. So this is only the concept where I'm going to show you the what we have done with this tool. And then we incorporate these two immunomodulators at the same time to make them bifunctional. The last thing is that because we are injecting the live tumor cells into the patient, we want to have a kind of like a safety switch to be incorporated in these tumor cells. So whenever these therapeutic cells that have done their job, that can eradicate all the tumor cells, we can clear these therapeutic tumor cells because they are also proliferating, highly proliferating. They also can grow themselves. We don't want them to become, and then they are already resistant to our therapeutics because it's by design, they are resistant. So we don't want them to stay. So we implement this safety switch using the HSV-TK suicide gene. So whenever you inject this canciclovir, the drug, into the body, that can activate this kill switch on the therapeutic tumor cells, and then to eradicate these tumor cells. The best thing about this HSV-TK is that there's also a bystander effect. So when these tumor cells they die, they also have they will kill the adjacent tumor cells. So it's another therapeutic effect that we have. So this is the whole idea of our schematic. First, we turn the therapeutic sensitive tumor cells into resistant. Then we give them immunomodulators. One can activate the programmed cell death, and then another one can modulate the immune responses. And then we also incorporate the safety switch that can kill the therapeutic tumor cells. At the same time, when the therapeutic tumor cells die, they can have the bystander effect to eradicate the adjacent tumor cells as well. And then after the tumor cell death, they will expose more tumor antigens, and then they will have the immunological memory. So that's the whole idea of the bifunctional therapeutic tumor cells that we have. Now I was going to show you some data. So this is the mouse data that we try to prove the preventive effect for these therapeutic tumor cells. So what we have done is that we first inject these therapeutic tumor cells to the mouse. So because in the mouse model, you can decide when they want to have, when these mouse are going to establish the tumor. So we can do this in the mouse model, but we cannot do this in human, of course. But we can do it in the mouse model. We inject the therapeutic cell to the mouse in the mouse brain, and then we see whether they are going to have tumor or not. So if they reject the tumor, we will re-challenge them with the wild-type tumor cells, that not the therapeutic type, and then see whether these wild-type tumor cells can still grow in the mouse. Does it make sense? So it's kind of like trying to see, it's a vaccine, to vaccine the mouse first so that when they encounter the similar type of tumor cells, they will reject it. So this is our data. So basically, we try with different groups. This is only the CRISPR tumor cells, only CRISPR after the knockout of interferon alpha receptor. We also have this GFP-TC. So they knock out, but they're expressing this GFP as a marker to see. We also have the single immune modulator therapeutic cells, GM-CSF and interferon beta, and the THTC with two together. And then as you can see that with the single, some of the mice that reject the tumor, so they don't grow. They grow a little bit, then they don't grow. And then only the double one that can eradicate, they're not eradicating, they're basically, you implant, they don't grow. They just become something that our immune system will target them. And then from these survived mice, these cohorts of survived mice from these three groups, GM-CSF only, interferon beta only, and THTC, we re-challenge them. We re-challenge them with the wild-type tumor cells. As you can see, only the double one that have complete zero tumor growth from the re-challenge, which means that we managed to vaccinate the mice to reject the tumor growth. We also implant some naive mice to make sure that our therapeutics, our implantation works, and all the naive mice that have the tumor. So which means that these tumor cells can grow, but if you already pre-vaccinated them, they won't have the tumor growth afterwards. And then, but we want to know what happened to these mice. So we use the immune-compromised mice to see. We use the not-skip mice. We inject these therapeutic cells on the not-skip mice. So you can see that without GCV, this tumor will keep growing. So this tells us that the mouse immune system itself plays the role to eradicate the tumor. Because when the mouse without the immune system, these tumor cells can grow. And then we further diagnose to try to see what kind of cells that are mandatory for this tumor growth inhibition and immunity. So we use the depletion. So we use the antibody depletion. We inject some antibody to deplete the CD4 T cells or CD8 T cells or both together. So you can see that when with the single, when you deplete them, only when you deplete double two types of the T cells, the mouse will have the tumor. As long as you deplete one type of the T cells, the mouse won't have the tumor, meaning that both CD4 and CD8 play a very important role to prevent the tumor from growing. And then we challenge those mice that survive from this to see which one that play the immunological memory, the role of the immunological memory. So you can see that the important role for this is the CD4 depletion. So if you deplete the mouse from the beginning with the CD4, even if the mouse didn't have the tumor growth, they won't develop the kind of memory toward the mouse. So if you re-challenge them with the wild-type tumor, the mouse will start to grow again after 2 months old, because we usually re-challenge them after 60 days, after the tumor, after the vaccine is gone, then we re-challenge them just to prove that it's a long-term memory. But if you deplete CD8 alone, it won't have the mouse will kind of still develop the immunological memory. So in this case, CD4 is the one that plays the role for the immunological memory in our case. So these are for preventive. We just want to show, because that's kind of like the protocol. If you want to say that this is a cancer vaccine, you really need to show the preventive effect. And then now we want to show the therapeutics effect of this THTC. So we are using the clinical relevant model. So what we did is that we first implant the tumor cells in the mouse brain. So when they establish the tumor, we did the resection. we basically do the surgery. That's how the GBM patient being treated. So when they have been diagnosed with GBM, the first thing is to do the resection to get the surgery. So we did the same thing. We resect the tumor. Then we put the therapeutic cells within the resection cavity, and then we check whether it's going to have any therapeutic effect or not. So you can see that we also have different groups. If you only have the single one, the control or the single GM7 or single interferon beta, they won't be able to eradicate the tumor. You can see that for the THTC, yes, we have the kind of like a very strong therapeutic effect. Although you still have some mice die, because just imagine this is a very aggressive tumor, but we have about 80% of the mice survive from the whole process. And then we also re-challenge these survived mice. They still reject the tumor. This tells us that if you use it as a therapeutic cells, they can also have the vaccine effect to vaccinate the mouse that has the potential to prevent the recurrence. Because for GBM patient, usually they die because of the recurrent. Recurrent is that the secondary tumor derived still coming back from the brain itself. Because as we can understand, when we do the surgery, it's not possible to clear all the tumor cells from the brain. So the best thing is that we clear as much as we could, because there's healthy cells. If you clear too much, basically this patient, they cannot function anymore. The well-being of the patient will be very high. So if we have the kind of vaccine that can prevent these tumor cells from coming back, that would be the best thing that we could do. So this kind of like very promising to us, because the therapeutic can actually prevent the tumor from coming back. We also did some analysis, the RNA-seq analysis, to show that what kind of this is a flow cytometry. This has been done for maybe 5 years ago. So to show that what kind of cells, that immune cells that after this therapeutic, we put the THTC, how they affect the tumor microenvironment. So as you can see, just briefly, you can see the increase of the traffic of leukocytes, the immune cells to the brain. Because brain is kind of immune privilege. Within the brain, there are very little amount of immune cells. Apart from microglia, the rest of the immune cells, T cells or macrophages, they're very little. And then that's because of the brain privilege. So if you can have the increased traffic of the immune cells going to the brain, it's kind of promising in this case. And we also have an increase of mature dendritic cells, kind of telling us there's an increased antigen presentation happening. We also have the T cells and also the CD4 T cells and CD8 T cells within the tumor microenvironment. We also run the immunofluorescence to validate the data, and then it supported what we found from the flow cytometry. And then we also did using the same strategy to try with the patient-derived cancer cells. This is only in vitro study. We tested 4 different human-derived GBM cells and then treat with our THTC and then see how it goes. So you can see that all of them, they have the therapeutic effect that we can see from here. If you go longer time, they're even better in this case. We also did a pilot experiment using humanized mice. Humanized mice is the mouse that we implanted. You basically grafting the human tissue on the mouse. We used the BLT mouse with the bone marrow and then the thymus and also the liver part of it, so that they can have full human immune system. Because some of the humanized mice, they only have maybe human T cells, but in our case, we have the full immune system of the human in this case. And then we tested this strategy with the human GBM cells, and then to see, because if we cannot use the human GBM cells in the immune competent mice, they will reject the tumor. The only way is to use the immune compromise, but then we cannot see the immune effect of our therapeutic cells. So the only way is to use the humanized mice that they don't have the mouse immune system that can accommodate the human's immune system, and we can also see. But it's very expensive. It's $1,000 per mouse. So that's why we only run a couple of mice in this case to show as a proof of principle. So from here, we kind of like have this conclusion that it can treat and prevent the GBM. But we also try to check with the brain metastasis whether it can prevent. So we try with the lung, because for lung tumor is actually curable. But if the lung travel to the brain, metastasize to the brain, it becomes incurable. So in this case, we want to see whether when we manage to detect the lung tumor that they are in the primary stage, whether we can vaccinate the mouse and then see whether they can prevent the brain metastasis. So in this case, we managed to show that some of the mice that can really prevent the brain metastasis in the lung tumor. We also did the same thing for melanoma. So we managed to, we inject the therapeutic cells of the melanoma cells in the mouse, and then we challenge the tumor in the brain and also in the for the skin, which is subcutaneous, and then see whether they develop the tumor. Apparently, they can prevent the distant metastasis in this case. So these all are promising. This is for this paper, and then but now I'm going to move on to share with you what we are currently doing, the progress of our work. They are promising, but we still have different challenge. The first challenge is that in the previous paper that we described is actually autologous strategy, which means that when the patient has the GBM, they did the resection, then we get the tumor cells from the patient, then only we can use those tumor cells to do the engineering to make them the bifunctional tumor cells. So in this case, the only chance for us to treat them will be the recurrent time, because then only they can open, because when they have recurrent, they will open the brain again and then do the surgery. That's the time that we can treat them, which means that we will miss the first chance of treating the patient in this case. So we want to avoid that. So that's why we are working on developing the off-the-shelf allogenic therapeutic tumor cells. So this is more into, you get the different tumor cells from the patients, and then you have them ready-made so that you can do, I'm going to describe them in details after this. And then so that you have a ready-made product. When the patient have the first surgery, you already have something to treat them. And then you can still get the patient tumor cell afterwards to modify them to get ready for recurrent. So in this case, we have two chances of treating the patient. So which is better strategy? There's another limitation is because we are using live therapeutic tumor cells. We are not using the attenuated. So in this case, the safety will be a big concern to the even for FDA or to the patient itself. We are putting some living tumor cells to my brain. So I don't know what will happen to me. So maybe the patient will have the concern on that. So we have two strategies doing that. One is to have the safety switch that we mentioned before. So in the previous paper, we incorporated two safety switch, the double Q switch in this THTC. And then after that, we also developed another Q switch that can better synergize with our therapeutics. And the other one is using the EV-based delivery. So I'm going to share with you for these three things that we are doing. So to have some rationale why we are thinking of using this off-the-shelf allogenic, we did some analysis analysis on the different type of murine tumor cells. So we found out that they're actually sharing different kind of mutations, different kind of antigens, about 88 of antigens, tumor antigens from these four different murine mouse tumor cells. Although they are different tumor cells, but they're sharing these 88 of them. And then we kind of like validate it to the human GBM. And then we use this 88 of the antigens to check on the human GBM. As you can see, surprisingly, a lot of them actually highly expressed compared to the normal tissue. This human GBM, they also highly expressed on these genes. So this kind of like telling us that we can actually develop something that have the shared antigens that can induce the immune response to target these tumor cells. We also check on different kind of a subtype of the GBM. That actually for example, we only use these three examples. They are kind of like sharing different, the same overexpression of these antigens. So we did some preliminary in vitro study. We are using these RNA S cells. This is actually a leukemic cells, very leukemic cells. They have a very kind of like special characteristic. So usually these cells, they don't have the expression of MHC1. They don't have expression in the normal room temperature. But if you, no, in the body temperature, 37. But if you put them in the room temperature, the MHC1 will be expressed. So we use this characteristic to validate whether these 88 of them are actually immunogenic. So how it goes is that when these peptide bind during this room temperature, they bind to this MHC1. When you put them back into the body temperature, 37 degree, they won't be able to retract. So this MHC1 will keep expressing. So this tells us that they are actually holding the peptide itself. So to tell us that it's whether they are a strong binder or a weak binder. From among these 88 candidates, we choose about nine peptides, and then we did the analysis on that. So you can see that within these 8 peptides, some of them are very immunogenic. They are comparable to OVA, which is a known immunogenic antigens, and also HPV, also a known one. They're basically comparable. So this tells us that this strategy could work if we manage to use them as the antigens. So the whole idea is that we get the tumor cells from different patients, because different patients, they have different characteristics. They might share different antigens. And then we modify them to make them the THTC. In this case, we knock out the interferon alpha receptor 1. We incorporate GM-CSF to them and interferon beta to them. We can also put the SCFP-PD1 is an anti-PD1 to them if we want, because since they are tumor cells, you can engineer them multiple times. Then from this, we use this kind of like mixed allogenic tumor cells as a therapeutic to put them when a patient being diagnosed with the GBM that did the first surgery, during that time we can use these tumor cells, the mixed allogenic of the cell tumor cells as a therapeutics. So to really make it happening, we are developing the mouse model first in this case. So we use different GBM cells from the mouse. They are from different backgrounds. One is from VMDK, one is the B62, F1 and GMM models to target the tumor from C57 black 6 months. Does it make sense? Because allogenic is that. So usually these 3 tumors, they won't grow in the C57 black 6 months because they are genetically different. But what we want to do because usually that's also what we are trying to do. Because if you inject the allogenic tumor cells into the patient, most of the time they will reject right away because it's a foreign cells to the patient itself. So this model can tell us whether our strategy is going to work. So this is a murine allogenic THT cell. And then we did some preliminary data. So we modified this SMA 560 to treat the CT2A. CT2A is the one that I say the GBM with the C57 background mouse. And then you can see that these different backgrounds, they can still kill the tumor cells. And then when we use the knockout CT2A with the knockout interferon alpha receptor, we reverse the effect. They won't be able to kill the tumor cells here. So which tells us that we can really get the tumor effect, killing effect through the interferon alpha receptor 1. But this is work in progress. We're still working now. I just did the in vivo experiment. Now I'm still waiting. My colleague is actually taking care of my mice to see how it goes. So we already modified 3 different GBM cells. Now we treat the CT2A cells. And if it works, so we are going to move to the patient-derived lines. And then another story that I was trying to say is that because of the safety concern, so we developed two different double Q switch. One is the GCV that we mentioned, HSV-TK. When you give the ganciclovir, they will induce the cell death, and then they will have the bystander effect. The other one, but this system will take longer. It will take about 3 days to really see the effect. So we developed another fast Q switch that when you give the dimerizer, it can induce the dimerization of the caspase-9. It can right away induce the cleavage of caspases and then induce the apoptosis. In this case, when the apoptosis happening, you will have the antigen shading. Although this caspase-9, Cas9 Q switch, they don't have the bystander effect. They don't have. So in this case, we combine 2 together, then we can avoid, because tumor cells are very, they can mutate they can evolve. So if we have two safety lines, then we can avoid any changes within the body of the patient. And then we also develop another type of Q switch, which is inducing the RIPK3 that can have the dimerization of the MLKL to induce the necroptosis. So what's the difference between apoptosis and necroptosis? Apoptosis is a kind of polylogenic cell death. So when the cells die, they will release a kind of signal to tell people that, to tell the immune system that this is a don't eat me signal. They will release don't eat me signal because our body always doing apoptosis. So down the road, because our ultimate goal is to train the immune system to target the tumor cells, to learn that they are foreign. So if we use the apoptosis mechanism, it's kind of like reverse the effect. You are telling them, don't eat me, don't eat me, basically telling them this is our tumor cells. This is different with necroptosis. When this necroptosis is happening, they will release the eat me signal. Come and eat me, come and eat me. Together with the danger signal. Then they can tell us that, they can tell the immune system, these are the foreign cells that you should target. So down the road, you can increase the effect of the immune system. So that's why we are more keen to develop this system in our research now. So this is why we want to activate, because we want to have this suicide gene system to have a synergistic effect to our immunotherapy. So you can see that they have bystander killing, they have this immunogenic cell death that can have the immunostimulatory cytokines, which will actually help us to target the solid tumor better. We actually published a review paper last year about this, why we want to incorporate suicide gene system in the cell-based therapy. And as I mentioned, we focus on RIPK3-induced necroptosis Q switch. So basically, this is the whole idea. We have the GBM cells, we engineer them to express this RIPK3 suicide gene, and then we can activate them with the BB homodimerizer. After they have the therapeutic effect, then we can induce them with the BB homodimerizer to induce the necroptosis of these therapeutic cells, which can further release the tumor antigens and promote the antigen presentation. Then ultimately, it can kill the tumor cells better. So this is a synergistic therapeutic effect that we are trying to have. So some data to prove that it works compared to two different, RC9 is the rapacus9 Q switch, and also TK is the HSV TK. Compared to the RIPK3 and rapacus9, they both effectively eradicate the therapeutic cells in the mouse. But then the next thing that we want to see whether they can induce the immunological memory or not. So from these surviving mice, we challenge them with the wild-type tumor. Apparently, only the one that with RIPK3 that can have the immunological memory. In this case, we are not using any therapeutic yet. It's only the tumor cells with the Q switch. We just want to purely see how immunological effect of this Q switch itself. And then we also try to incorporate them in the therapeutics. The same thing that we did is that we implant the tumor cells and then we intratumorally implant the therapeutic cells with interferon beta and RIPK3. And then if the mouse survives, then we will challenge them and then we monitor them. So from here, you can see that therapeutically, when we activate the Q switch, some of the mice will survive. And then none of the mice with interferon. We on purposely didn't include the GM-CSF because in the previous study, we know that if you have two interferon beta and GM-CSF together, they will have the therapeutic effect. So we don't want to have a clear therapeutic effect from here. Otherwise, we won't be able to see the effect of the Q switch itself. So in this case, we on purposely left out the GM-CSF, only leave the interferon beta here. Then from here, we can see that if you only have interferon beta without the Q switch, the mouse will die of the tumor growth. But with the Q switch, they'll survive. And then we challenge those mice, they have the immunological memory. So we also try with these two different models, CT2 and GL261, another GBM models, to see whether they have the same effect. And then we can see that they're basically doing the same thing. So this tells us that if we incorporate the safety switch, we can synergistically enhance the therapeutic efficacy. but this is still work in progress. Now, this also published in 2025 at JCI, but now we are trying to move, because this is still the Q switch itself. We want to make them the stand-alone therapeutics, as the repatriate as the stand-alone therapeutics. We are trying to develop the mRNA with the LNP hydrogel as a stand-alone therapeutics instead of only using the cell-based therapy. The next thing that I'm talking about is about the EV, the extracellular vesicles. The focus is the tumor-derived nanovesicles. I call nanovesicles. So we know that the living tumors, they are alive, they can proliferate. But by using the extracellular vesicles itself, they are organic, but they are not living. They cannot proliferate. So which is an advantage for us because they are natural delivery system. This tumor-derived EV, they have the same cell homing capability. This EV, because when the tumor derives this EV, is for the communication between the tumor cells. So when you use this tumor-derived EV as a therapeutics, they can actually also travel back to the tumor cells to communicate at the same time to eradicate the tumor cells. And then they also have, the good thing is that they also have a lot of tumor antigens within the EV itself. And they also have the targeting abilities. So you can see that our whole strategy here, the lab is trying to make the tumor cells as a spy, as a turncoat, as something that can target the tumor itself. So in this case, in our tumor-derived extracellular vesicles, we are doing the same thing. How we are going to do that is that we basically, first, we use the tumors. We are not modifying the EV itself. We are modifying the tumor cells that are producing the EVs. So we knock out the interferon alpha receptor from the tumor resistance, the same thing. And then we make them we also make them to express the interferon beta. Instead of expressing the interferon beta as an agent, we make them bind to the EVs. It's a membrane-bound EVs. So this EV, they have the interferon beta with them traveling so that they can target the tumor cells. And then in this case, we have the antigens. We also have the interferon beta. They will play as a bifunctional effect in this case. So the whole idea is that on the EV, there are different kind of protein that you can use. So we basically use the engineering to make the interferon beta bind to this lactoherin C1, C2. So they will become the membrane-bound interferon beta TDN, the extracellular vesicles. So from here, you can see the control tumor-derived EV that don't have the expression of interferon beta. But from here, we can see that with the MITDN, you have the expression of interferon beta. And then this is just the proof that they are the EVs. And then we try the cell viability. We use this EV to treat the CT2A to see whether they can treat. So you can see that they can kill the tumor cells. We also try with the EV derived from CT2A to kill other murine tumor cells. Although it's not as effective as the CT2A, but yes, they can still kill the other tumor cells, although this EV is derived from CT2A. We also try with the mouse model. So we intracranially implant the CT2A, and then we intratumorally treat with the membrane-bound interferon beta EV and see how it goes. So you can see that improved a little bit of the therapeutics effect there, although it's not very promising. And then they also increase the maturation of dendritic cells. We are actually trying to make it better. Now we are trying to develop another strategy on this. So this is still ongoing. This is more for the therapeutic effect. We are also looking at the preventive effect. What we are doing is that the time is running out. We ex vivo culture the splenocyte. We collect the splenocyte from the mouse. I promise this will be the last few slides. We extract the splenocyte from the mouse, and then we ex vivo culture them together with our tumor-derived EVs with the control. And then 2 days later, we collect the splenocyte. We co-culture with the tumor cells, and we always do the flow cytometry. You can see that the treated splenocyte with the MITDN treated splenocyte can kill the tumor. So this basically tells us that the immune cells being trained by the membrane-bound interferon beta derived EVs that can treat the tumor. And then we also noticed that some maturation of CDC1 and also the increase of the T cells. And then we also tried in vivo. So what we did is that intramuscularly, because usually vaccine is injected intramuscularly, inject the PBS or the MITD and our membrane EV. In this case, we didn't use the control because the control EV, they are still kind of like tumor-derived. They will introduce the antigens. To have a clean strategy, we didn't introduce that. 3 times and once per week for the 3 weeks. Then we collect the sphenocyte. We stimulate them with the peptide that I talked in the beginning that can work with the SMA S cells, and then we quantify with ELISA sport. So you can see that they can really induce the immune response, make them more vaccinated, the immune cells in this case. So this is still ongoing. In conclusion, we have this whole mechanism. We developed this interferon beta plus GM-CSF THTC, and then they have the direct killing, they have the immune activation. But we are facing 2 different limitations, which we try to solve with the off-the-shelf allogenic THTC, so that they have readily available, scalable production and consistent quality. And we have the safety layer for the safety concern to have the suicide gene or use the non-living version of the THTC by using EV. So by doing this, we hope that we can move this forward to a you can see that we are trying to mimic, we try to use things translationally so that we can see how it can go to the patient. When we design our experiment or design our project, we always try to think like that. Is it possible to go to the patient like that? If it's not, how we can do that? How we can make it better? Then we only do that version that can, the likelihood that we have a high likelihood that can go to the patient. So that's why we didn't focus on going very deeper and deeper until we found the solution, then we go to that. And then, oh, it's already time. Give me 2 more minutes. And then because Murni gave me some hints that most of the audience is more plant-based, then I tried to think of what kind of things that we can do with the plant-derived drugs that can plug into our platform. So I tried to think of the difference between the plant-based therapy and cell-based therapy, chemical living system, programmable, broader targets. And then the thing is that PK-driven clearance would be some key issue for the plant-based therapy. And then we will have the more persistent because you're using cell-based, but you need to control it. There's always the downside of that. Manufacturing usually is easier because you're dealing with more something that you can really control more. And delivery is more to the problem that we have is the bioavailability and penetration. Because the batch of the plant-based therapy could be different. So QC might be tougher for that. And for the cell-based therapy will be the viability, trafficking, and compatibility. And then I try to think of how I can incorporate the plant-based therapy into my strategy. Because the good thing is that using with the cell-based therapy, they can always incorporate different kind of therapy. You can have drugs, you can have chemo, you can have surgery, you can have radiotherapy. That's the best thing. Although we try to avoid surgery, chemo, and also radiotherapy because they are too damaging, too toxic to the patient. We try to avoid that. So we always try to choose something less toxic. Plant-based could be something very interesting. So 3 ideas. One is to prime the tumor microenvironment. Before we use the cell-based therapy, we can use the plant-based therapy to prime the immune tumor microenvironment first, and then or to boost the EV-based vaccine potential. So one thing that is that we can encapsulate the EVs better using the plant-based system, or to improve the formulation and delivery in this case to make them the biocompatible strategy. So with that, thank you for the long presentation. I didn't expect that. I just tried to. I thought it's a very short one. So these are our group. So our director is Dr. Karisha, and then we have now this is kind of like this was taken last year during the Christmas dinner that we had, and then yeah. So basically we are open to any collaboration, especially as what I mentioned here. If you have any idea, so please reach out to Murni, and then she will see how we can work together. That's also my goal, and because I feel like the best thing to bring the technology from other countries is to have collaboration. because you can use the resources from other countries. At the same time, you can bring the technology and knowledge to our home country. So thank you. If we still have time for questions, then I can take some questions. Thank you.
Okay, thank you very much, Dr. Chen Siong, for the very insightful and provoking presentation here. It's quite advanced. You can see how advanced is Harvard as compared to other universities. But yes, we are open for collaboration so that maybe we can learn something from the institution. Okay, now let's open for question and answer session. So we have one question from Hospital Cafe. Let me just read so that both parties can listen.
This one?
Okay, the first one. From Hospital Cafe. I would like to know if this applicable under study to giant cell tumor in case that has spread to lung cells. Patient is currently under morphine already. Left BKA done a couple years ago. So what do you think?
So in theory, this strategy should also work for any tumor types, as long as they are easy to engineer, and then they have this interferon alpha receptor as a target. So the first thing, if let's say with this strategy go to the patient, the first thing is to stratify the patient. We will screen with the interferon alpha receptor because that's the main killing agent that we have. Then from there, we can see if they are overexpressing interferon alpha receptor 1, meaning that this could be a good target for us. If it's not, we also have other type of therapeutic agents. For example, our center also working on TRAIL. TRAIL is another therapeutic agent that can induce the apoptosis binding to the DR5 ligand, the DR5 receptor, which is a death receptor of the tumor cells. So there are always strategy that we can choose what kind of therapeutics. That's the beauty of that, because you can engineer. Of course, for the off-the-shelf strategy, we will stick to the interferon beta first. But down the road, what our aim is that we can build different kind of off-the-shelf products that can be suitable for the patient. Maybe not directly to explain because I'm not a clinician, so I'm not really able to go through the patient part for that. But from the scientific point of view, yes, definitely. As long as we can get the tumor cells, we can engineer them, then we should be able to make them a kind of like cancer vaccine that we want. So this works very well for leukemia patient, because the cell homing itself is so effective in this case. But our lab focus on GBM, we try to target the most efficient, the most aggressive one first. Then we ultimately will go to the more easier targeting. Lung cancer, as mentioned, that in our case, we can also use the tumor cells from the lung to modify them. They still have the same effect. And we also did the TCGA analysis on the mixed cancer type. Almost all the cancer types has this interferon alpha receptor 1 highly expressed on this. So we are kind of confident it can be applied to other tumor, but we will need to do more literature.
Okay, we have any question from the floor? Any? Okay, now we move to the second question. May I know the effect of THCC to B cells?
So very good questions. So our model in the so from the flow cytometry, we also look at the B cells, but basically we didn't see any changes of the amount of T cells of the B cells itself. So that's why we didn't really go into deeper on that, because if let's say there's a changes of number or amount, we might need to use different markers to really go into deeper. But it would be something interesting to know in this case. Our main focus is still the antigen, the adaptive pathway from the antigen presentation, and also the T cells and how the dendritic cell works from there. But it's definitely interesting to look at the B cells ultimately. So the short answer is that we didn't see any effect, but if we go deeper, maybe we might. Thank you.
Okay, perhaps one last question from myself. So how far are you from materialize this kind of treatment to the patient? Are you still going to go from clinical and then regulatory things? How far are you?
So that's a very important question, because you can see that most of our work is preclinical, but the way how we think about is for the translation. So that's why all our models is more clinically relevant. So we prepare ourselves from the preclinical model to the clinical model. So we are already in the discussion with FDA. We already have the first meeting last November with them to see how we can run the phase 1 or even the phase 0 clinical trial with 1 or 2 patients, because GBM is very aggressive. So most patients will want to try for that. So that's why when our paper published, a lot of patients kind of contacted us. But unfortunately, that time our strategy is still in process. There's no product yet. But now we are already preparing the cells into a GMP grade. We already have the stocks on that. So we just need to get more investors to invest in the company, in the startup company, so that it can fund the phase 1 clinical trial. But I guess it will take at least another 2 to 3 years to be able to test on the first patient. So that's a very long timeline. So to me, it's definitely very promising. We already kind of ready for that, but it's all up to the FDA. And last year, the government temporarily shut down for a few months, so this kind of also delayed the process. But it's all about money also, because without money, you cannot do clinical trial. In this case, unless the hospital wants to fund it, but most of the time you will need to find investor to fund your startup to really run the clinical trial. I think that the biggest bottleneck usually is because of that. If you are very good businessman, you will get funding easily, even your research is not very strong. But if you are not, then you really need to use your data to really support that. Does it make sense? Thank you.
Yes, I think still have many stages to go, right?
Yes.
All right, thank you very much, Dr. Chen Siong. Let us give another question?
Yeah.
Oh, sorry. Maybe last question, I think. Is there a possibility to give the cancer vaccine to healthy people to boost their immune system against cancer? Like vaccine.
Well, that's why I don't think people. I mean, we can do that, but I don't think people will want to do that. So, for example, I have a son. So even for the commonly known vaccine, I also still have a doubt whether at his age, because in United States they have the standard procedure to give maybe 5 vaccine at one time. We always say that can we delay a little bit, because you cannot. It's kind of hard to see that it's not because of the doubt of the vaccine. It's to poke 5 times at once. Like you can see that the baby is still only 6 months old. You already poke them multiple times. So that's also one thing. Is the concern of the normal healthy human. Are you okay by injecting proliferating cancer cells into your body? I don't think people can accept that. To me, I also cannot accept that, because what will happen to me? Although I already have this kill switch, but I'm healthy. So which is hard to convince people. But when people is sick, they already don't have any other option, then that will be the kind of the lifeline for them to really pick it up and then to hope that they can help. So yeah, basically, I don't think this will be something that the common vaccine that we can do, but mRNA vaccine maybe. mRNA-based vaccine. So this is also another arm of our research, because mRNA vaccine is very popular now. We also use our analysis, based on our data from the analysis itself, we try to develop the mRNA kind of vaccine. In this case, we can with mRNA vaccine. You can, for example, GBM, there are multiple different kind of antigens that already are known. We can combine them together with other to boost them, and then you pre-inject to the people that prone to tumor. For example, the P53 mutated patient, yes, personals or something like that, like Li-Fraumeni syndrome. They have the Li-Fraumeni syndrome. Then in this case, we can kind of use mRNA vaccine to them. But to put the cancer cells, in short answer, I don't think.
Yes, so the answer is no. All right, I think that's the end of the seminars today. So before we end, can we give a round of applause to Dr. Chen Siong? All right, thank you very much, Dr. Siong. Okay, so now can I call upon our director, Professor Dr. Shahrul Makarum, to present a token of appreciation to our speaker, Dr. Chen Siong. Thank you.